So let's start with a simple definition of what we mean by heterozygosity. At any single locus in the genome, we have a choice of 4 different base pairs. We've got GC, AT, and since the order matters, CG is not the same as GC, TA is not the same as AT. So we've got 4 different choices at any given locus. We've got 2 chromosomes in a human genome, in a human cell, one from the mother, one from the father. If they have the same information, then they're going to be homozygous. So we've got guys down here where we've got GC base pairs on both the maternal and the paternal copies at a particular locus. Or if they have different information, they're going to be heterozygous. So here's guys, I got an AT base pair from one parent and a GC base pair at that locus from the other parent. That's heterozygosity. This down here is homozygosity. The term allele just refers to what information I have here at this locus. So traditionally in genetics, instead of calling them their base pair names, because we didn't know about base pairs, is we name them A or B. So I'm going to call this allele A and this allele B, just randomly. That doesn't say anything about what information is encoded at that locus. I'm just going to call the first one A and the second one B. So at this particular place, I have both A information and B information, and I'm heterozygous. Down here, I have just B information, and I'm homozygous. Double-strand break repair can cause me to lose heterozygosity, which means I either become homozygous or I lose a copy of the information and I become hemizygous. So this is an overview of the different ways I can lose heterozygosity. We'll go through each of them in separate slides after this one, but this is an overview. Okay, the way things are supposed to go is I'm a diploid cell over here, and I've got a copy of each chromosome from my mother and a copy from my father. And so I've made them blue and green here, and I'm looking at just one chromosome. But of course, the human cell has 23 pairs. When I go through S phase, I replicate these, and now I've got 4 copies of each chromosome. What I'm supposed to do at mitosis is I'm supposed to take one copy of the paternal chromosome and send it off in one direction, and send the other one off in another direction. I'm supposed to take one copy of the maternal chromosome and send it off one the other way, so that the 2 daughter cells each get one copy of the maternal and the paternal, and I end up with 2 diploid cells that each have one copy of the maternal and the paternal. So that's the way it's supposed to go. But if something goes wrong, as here, I get DNA damage and I get a double-strand break down here in one copy of the chromosome. So the DNA damage is supposed to signal for repair. One way that can be repaired is through homologous repair mechanisms. Down here, the 2 ends of that break invade the homolog. During the resolution of that homolog, I can either resolve that to give crossing over, as is shown here, where now the maternal and the paternal arms have been exchanged, or I could resolve it so that I just get patch repair and I won't see that. So we're not going to consider that case. We're just going to consider the case where we get crossing over as a result of the homologous recombination. So in that case, now the centromeres at the next mitosis are going to do the right thing, and they're going to send one centromere off this way, one that way, one this way, one off that way. And except now, when the centromeres did the right thing, they've got a maternal and a paternal copy in each of the daughters, but we dragged along the distal arm of the broken chromosome so that now out here I'm homozygous and I just have the blue information. Down here I'm homozygous and I just have the green information. So now everything distal to the event where the double-strand break was and where the crossing over occurred is homozygous, which is to say I've lost heterozygosity. I'm now homozygous at all of those loci. Another way that can happen is, again, I have my double-strand break down here and one copy of a chromosome, except now the checkpoint fails and doesn't say, hey, you guys, let's slow down and fix this, so no repair event occurs. And then during the next mitosis, again, the centromeres do the thing they're supposed to do. They send the two copies of the chromosomes off in their separate directions. But this little fragment wasn't attached. It didn't have a centromere on it. It wasn't joined to a centromere. So it stays behind at the mitotic plate and gets lost. As the two daughter cells form, it's outside the cell. So this guy got a perfectly normal genome, but this one ended up without any of that information out there except the one copy it inherited from the blue chromosome. So since it can't be heterozygous, it only has one copy of the information. It can't have both things if you only have one copy. It has also lost heterozygosity. It isn't homozygous, it's hemizygous. There's only one copy, so we don't call it homozygous. But we have once again lost heterozygosity distal to the point of the double-strand break through a different mechanism.